one thing when you run a drug trial for pharmaceutical drugs is that many patients are dropping out because of the side effects. But for brain stimulation, the patients are so eager that they basically come back for more brain stimulations. And you don't see that in drug trials due to the side effects. It looks like a headphone. So you see the transparent bit, which is just fixating it in the ear. And the blue bit is emitting ultrasound. The ultrasound is activating parts of the vagus nerve that is situated around the ear. And this vagus nerve stimulation is then activating the parasympathetic system. You have a wide range of benefits, people sleep better, so you use that device for 5 minutes a day, and then after a week, anxiety is already down by more than 30% and then down by more than 50% after a couple of weeks. Beyond the next 5 years, I would think that this will change clinical practice. So there was one psychiatrist who basically said that in the future, every psychiatrist will have a sofa in his office and a brain stimulation device.
Thank you for joining Once Changing the World, India's first future tech meets sustainability podcast. I'm your host, Eddie Aaron. Today I'm delighted to welcome a leading expert in neuroinformatics and brain connectivity, Professor of Neuroinformatics at the University of Nottingham School of Medicine. He is the author of Changing Connectomes. His leadership roles include Chair of Informatics UK, Chair of the Neuroinformatics Special Interest Group at the British Neuroscience Association, and Chair of the NHS Chain Technology Subgroup on Computational Neurology. Professor Kaiser's research and work areas include network science, models of brain development, neuroinformatics, computational neuroscience, epilepsy surgery predictions, brain folding, brain connectivity, connectomics, and focused ultrasound with application in diagnosing and treating neurological and neurodevelopment disorders. And really appreciate you taking time and being part of the podcast. Your research focus is largely on the connectomics side. So it would be great if you could maybe start with explaining what connectomics is, the human connectome project, and maybe also if you can give an overview of your book, Changing Connectomes.
Okay. So the connectome is a total set of connections in the brain. And connections could be structural, functional, or effective connectivity. Structural connections are fiber tracts in the brain or axons between individual nerve cells. Functional connectivity means that two parts of the brain are active in a similar fashion. So if you look at the activity over time, there's a similar pattern for two regions. So if there's a strong correlation between activity, you would say there's a functional link between two regions. Or finally, you could look at effective connectivity. And effective connectivity means that one region is influencing activity in another region. So let's say one region becomes more active and then another region becomes more active or becomes less active. So if there's a causal relationship, there would be effective connectivity. So the total set of connections in the brain is called the connectome. And that's a term that just came up, initially proposed by Olaf Spons in 2004 and then used in his publication in 2005. You have all those different kinds of omics, proteomics, genomics, and you basically have connectomics, looking at how the brain and the nervous system is connected.
Right. And will be great if you could also give an overview of your book, Changing Connectomes, and how has the field evolved since you wrote the book, which was published in 2020?
Yes. So the field has really exploded. When I started to work on brain connectivity in 2002 during my PhD, there were very few people worldwide, so maybe a dozen researchers worldwide working in the area. Nowadays, if you go to a new science conference, if you go to a conference looking at new imaging, for example, MRI, most of the work is including some concepts from connectomics. So the field has completely exploded. There are now thousands of researchers in the area, whereas in 2002, there were only a dozen worldwide working in this area. And we are moving more from understanding how the connectome is organized in general in humans, but also for different brain diseases. We're moving to understand how the brain is changing over time. And this is what the book is about as well. So Changing Connectomes. The book is introducing concepts about how the brain is connected as a network and how those different features of the network originate, both during brain development, but also how they originate during evolution, what those features are good for, and then how those features differ between healthy development and new developmental conditions like autism, schizophrenia, depression, and how they differ for old age conditions like Parkinson's disease and dementia. And then finally, how we can change the structure of the connectome through learning and through brain stimulation.
So, Professor, this Connectome project, I mean, the human Connectome project, besides the human Connectome project, there's been like various efforts. There was a C. elegans, which was completely mapped because it's just 302 odd neuron. And then there's a fruit fly, which has been completely mapped. And then there's Google Harvard, which is kind of, mapped a cubic millimeter of the brain. Can you explain this process of the connectome? How do you go about it? It's this slicing of the brains, and then you kind of map it onto the computer. How does that work? And how rich is the resolution of this collected data of these connectomes?
Yes. So for C. elegans, it's based on electron microscopy. So you basically slice up the C. elegans. It's a round worm, around 1.4 millimeters long. You slice up the whole animal in small slices. You use electron microscopy to reconstruct the shape of individual cells. And then you can trace the nerve cell, but also the axon going from the nerve cell. So you can reconstruct how different cells are connected. And this took a long time in the 80s. So it's really 5 to 10 years of work to do this kind of reconstruction. So this is what we would call the microconnectome. It's detailed information about how individual cells are connected. And you can use some similar or related techniques when you go to the fruit fly Drosophila. But when you go to larger animals, it would take too long to reconstruct it at this level. So I was asking one colleague, how long would it take to reconstruct the human brain at the same level of C. elegans, so connections between neurons, and he said it would take 40 years. And 40 years is assuming that the technology is getting better every time. And it's still 40 years. So for the human brain, we don't have detailed information about how individual neurons are connected, but we do know how brain regions are connected. So you can split up the human brain into, let's say, 100 brain regions, and then you can observe how well those 100 regions are connected. This is what we call the macroconnectomes, not the microconnectome, but the macroconnectome, where we look at larger structures in the brain and how they are connected. So in order to reconstruct the macroconnectome, we can use a special sequence in an MRI scanner called diffusion imaging. So it's a standard MRI scanner you have in a hospital or in a research institute. You have a special protocol for a couple of minutes, and then we can reconstruct how different parts of the brain are connected. And based on this macroconnectome, we can already predict the age of a participant plus minus 8 years, or the intelligence, the IQ of a participant plus minus 3 to 5 IQ points. So just looking at the structure of the network can give us some information, even if we don't go down to the microconnectome level of individual neurons and individual axons.
Right. How interesting is that? And I mean, obviously 40 years seems to be a long time, but at the pace at which technology is growing, I'm sure maybe the time horizon might get closer. So when you say that, you're creating these connectomes, you're trying to structurally, functionally, effectively also, capture the entire data of like maybe like a human brain. Besides these age and things like that, what are the understandings? What are we learning more from these connectomics?
Yes. So one point is that the human brain, or in general, biological brains, are very different from computers. So even though there is artificial intelligence and there's the idea to get closer to the capacity of the human brain, the architecture is still very different. So one feature we find is a modular organization. So modular means you have many connections within a module, but few connections to other parts of the network. And this gives you functional specialization. So you could have one module for visual processing. So you have lots of interactions to determine what object is there in the outside world, but you're not interfering with sound, with auditory stimulation. So you have a specialization for different parts. And this is still different if we look at artificial neural networks. Artificial neural networks are good at one modality. So they're, for example, good at object detection. If you look at a CCTV camera, but they don't include sound and they don't include other kinds of information. So it's still unique for biological systems that you have those different modules that are processing very different kinds of information, but at some point they're all interacting to find out what is going on in the outside world. So modular organization is a big characteristic of biological connectomes. And you also have a hierarchical modular organization. So, for example, you have one module for visual processing, but then within the visual module, you have two smaller modules nested inside. And those smaller modules have further specialization. So one further module in the human brain is the ventral pathway for object recognition. So, for example, you recognize the color of a coffee cup and that it is a coffee cup. And then you have a different module, dorsal pathway, and that is about the position of the object. So where on the table is the coffee cup, and is the coffee cup moving? Is it falling off the table or something like that? So you have further specialization at different hierarchical levels. And that's really characteristic of biological connectomes.
Right. So, Professor, when you say you using these approaches, both, modular and hierarchical, what are the learnings from there? And are these models able to predict, this disease progression or treatment outcomes?
Yes. So one example is epilepsy. If you look at the network organization of epilepsy and you look at functional connectivity interactions between different parts of the brain, you see that epilepsy patients have stronger connectivity between modules. So modules normally would be bottlenecks for the further spreading of information. Information can easily spread within a module because there are many interactions within a module, but then there are few connections between modules. So it's difficult for activity to spread between modules. But for epilepsy patients, they have more connections between modules, so it's easier for activity to spread. And this is exactly what you see during epileptic seizures, that you have one starting point, but then you have large-scale spreading of epileptic activity in the brain. So there is this link between the organization of the network and what you see in patients. And we see other characteristic differences. If you look at dementia, or if you look at schizophrenia, we see basically network changes that can help to explain why those diseases originate and why they have a certain effect on cognition. So one example is for dementia, for Alzheimer's disease, we see reduced myelination. So myelin is wrapped around axons, and it makes the propagation of activity faster. Okay, so when axons are well isolated, then activity can spread at 10 times the speed compared to not being isolated. And we find a reduction in this isolation in dementia patients. So it takes longer for the information to arrive. The delays in the system are longer. And this could explain why the system doesn't perform that well anymore. So when you think about recognizing something in the outside world, you have, so let's say you have a face. So you have to recognize the face, you have to recognize what is the name of that person. So you have different parts of the brain that are interacting. But if the information doesn't arrive at the same time, then you can't combine the information. So if the delays are wrong, then information arrives at the same time. So one part of the brain tells you, oh, I know that person. And the other part of the brain tells you the name of the person. But because those two are not interacting at the right time, you don't match them together.
Interesting. Professor, you also have done work on optogenetics. Will you be able to explain first what optogenetics is and how are you leveraging optogenetics in stimulation for epilepsy?
So that was a 7-year project where we tried to develop novel optogenetic stimulation. The idea was to test it in human epilepsy patients on the long term. We didn't get to the human stage, but we did do tests in rodents and in rhesus monkeys. What you do for optogenetic stimulation is that you have an implant. So you have something that you stick into the brain tissue. And this implant is containing different components. One component is a normal electrode to measure information, so to measure what is going on in the brain. And the idea is that you measure ongoing activity, and based on that, you determine when should you stimulate. Okay. And another part of what you stick into the brain are very tiny LEDs. So they have to be very tiny because you don't want to heat up the tissue too much. Okay. So if you have a normal LED and you turn it on, then it would heat up the tissue too much. And you're not allowed to heat up the tissue by more than 2 degrees Celsius. So it has to stay below the limit. So the LEDs are very, very small. And the LEDs are shining light into the tissue, where the light is only traveling about a millimeter. So you need several of those units stuck into the brain in order to stimulate a large part of the brain. So what you do with optogenetic stimulation is that you first inject a virus into the tissue, and this virus is containing information that is changing the properties of cells in the tissue, of channels in the tissue. And because the properties are changed, some of those channels are able to open when you shine light on them. And this is the way how you can change brain activity. You shine light, and then depending on the channel, you can either increase or decrease activity in the system. So it's a very complicated process. You're first injecting a virus. You have to wait a bit until it's diffusing in the tissue. And in the second stage, you have an implant that is stuck into the brain. So even though the experiments were successful and we saw what we were expecting to see, we saw a reduction in activity in the system, it's a very complicated process, and it has led me and others to the idea that what we really want is something non-invasive, something that works without implanting anything into the brain.
Neurological disorders, it's like a huge challenge. There is like millions of people around the world who are suffering with deep neurological disorders, right from Alzheimer's, depression, schizophrenia, Down syndrome, and so on and so forth. The list is like pretty much very, very wide, but and there is an acceleration of tech, where there's with convergence of sorts, with neuroscience, with biotech, with neurotechnologies, brain computer interfaces and things like that. These tech stack are still in nascent stage, and these are still problematic. And I mean, we would definitely get into the non-invasive approaches, but the invasive approaches are the approach at this point in time which can capture proper resolutions and maybe create more impact rather than the non-invasive ways. We will get into the non-invasive, but I want to talk about the invasive approaches. What's the cutting edge of treatment at this point in time, with invasive approaches, which is showing benefits across the neurological disorder spectrum, from Alzheimer's to depression to Parkinson's to maybe schizophrenia and others. It would be great if you could talk about that.
Yes. So the most successful area at the moment is Parkinson's disease, where you have stimulators that are implanted, and then you basically have a reduction in tremor and improvements for patients with those implants. There was an area for epilepsy implants as well with electrical stimulation. There were cases where it is used for obsessive compulsive disorder. Fewer cases in that area. So the main area at the moment is still Parkinson's disease, and in general, any kinds of motor diseases where this is used. But even for Parkinson's disease, there is a limit on how many patients can benefit. The limit is, on the one hand, some exclusion criteria, but on the other hand, it's a limited amount of surgeries that can take place. So it's limited by the number of new surgeons that are trained in the procedure. And because of that, in the UK, for example, there are only 400 procedures every year in terms of deep brain stimulation implants. It's a higher proportion if you go for easier surgery like cochlear implants, but if you go for deep brain implants, it's a very complicated procedure and very, very few centers that can do it. In the UK, there are 12 centers, 12 hospitals that are able to do deep brain stimulation surgery and implants. So there was an upper limit on how many patients can actually benefit. So even if we would find hypothetically that it can be useful for depression or it can be useful for schizophrenia, so those are conditions that are affecting a large proportion of the population. There is not enough capability to use implants in that population, just based on the number of centers and the number of surgeons that are able to do it.
Right. I hope is that nanotechnology manages to reduce the size of these neurotech devices, and hopefully, I mean, we have like a mix and match of the non-invasive as well as the invasive. You're the one who kind of pushes the non-invasive approaches, and you are focusing on focused ultrasound neuromodulation. Will be great if you could talk about what is this focused ultrasound neuromodulation. How is it adding value?
We did a survey with the general public and patients. So 800 participants, half of them healthy, half of them had mental or brain health conditions. Many had Parkinson's disease, but we also had depression, schizophrenia, and other conditions. We were asking them, what kind of intervention would you prefer? So either for the patients, the condition they have, or for healthy participants, if they get a condition later on. We were asking about 5 different technologies. One is pharmaceutical drugs, which is the standard treatment for brain and mental health for most conditions. Then we had invasive technologies like implants. And then we had 3 non-invasive technologies. So non-invasive electrical stimulation, magnetic stimulation, or ultrasound. And the preferred option out of the 5, both for patients and for healthy participants, was ultrasound. People possibly just thought about pregnancy scans with ultrasound. So what ultrasound is doing, it's sending a sound wave through the skull and through the brain. And the sound source is basically curved. So it's called a transducer. It's a curved surface. So the wave is coming out of the surface and is then converging in one point. So basically, at this one point, the focal area where all the waves are converging, this is where you get the maximum pressure that ultrasound is generating, like as a mechanical wave. And the point where you have the maximum pressure, this is where you can change brain activity. So you can either decrease or increase brain activity at this focal area. And the benefit of ultrasound compared to other non-invasive technologies like magnetic or electrical is that you can directly target deep brain structures. So we know from for most diseases that deep brain structures are involved, like hippocampus, amygdala, thalamus, and so on, basal ganglia. And so you can go directly for those deep brain structures.
How interesting, because I think sound is so underexplored, because I personally am a musician. I used to kind of play music for the longest time, and I've always been intrigued by these Chladni plates or these cymatics, where you kind of have like a metal plate, and then you put these salt or sand, and you play a certain frequency, or you have like a violin, bow of a violin, and you kind of you try to get some certain frequencies, and these salt or sand, it kind of self arranges into these extremely elegantly beautiful geometrical patterns, so there is something obviously going on deep, and will be great if you can elaborate on this. You mentioned that through all of these approaches, the 5 approaches that you mentioned about drugs, and then invasive, and there's 3 non-invasive approaches, which is magnetic, electric, and ultrasound. Ultrasound seems to be the most beneficial. 800 participants. I'm sure there must have been some kind of a clinical results. I mean, is there anything that you would like to share on the benefits from this 800 participants, and what have been those benefits?
Yes. So the 800 participants, they were just filling in a survey about what they would like. But there were a lot of studies using ultrasound and other technologies, non-invasive technologies. It's a very safe procedure compared to others. So for magnetic stimulation, 30% of participants have some kind of side effect, like headache or some dizzy feeling and so on. For ultrasound, it's only 3%. So it's a very safe technology. So there was one result by colleagues in Paris, Jean-François Aubry. They were using ultrasound to target deep brain structures that you would normally target with an implant for Parkinson's disease. So they were directly going for those deep brain structures, and they were able to reduce the tremor in essential tremor patients by 98% or 99% for at least half an hour. So you basically have non-invasive benefits that you normally would only get with implants. There were pilot studies in other conditions. There was one study in schizophrenia in Shanghai published last year. And in our center in Nottingham, we just got a 5-year grant to really look at novel technologies of ultrasound and magnetic stimulation, where we look at depression and schizophrenia. And we also look at Tourette syndrome to see whether we can improve brain function with non-invasive technology. And the difference for the technology that we develop is that we are using closed loop stimulation. So open loop means that you just stimulate at a certain point, this regardless of the state where the brain is in, regardless of ongoing brain activity. And closed loop means that you have information about how the brain is functioning at the moment, and then you time your stimulation to be just at the right time. And we know from the Optogenetic project that I mentioned earlier that closed loop has huge benefits in terms of the effect on brain activity. So we aim to see the same beneficial effect in those human participants in depression, schizophrenia, and Tourette syndrome to have much better outcomes using our closed loop approach.
Wish you the very, very best for it, Professor. Has there been any accounts where this approach has been FDA approved? And what's the lab to market timelines that you're looking at?
Yes. At the moment, it's still early stages. So in terms of using ultrasound to change brain activity, to my knowledge, there has been no FDA approval yet. It's still the early stages of development. However, there are other tools that are on the market already. So there was one company where I'm on the scientific advisory board, NewGear, and they developed this device here. So let's see whether I can hold it in the camera. It looks like a headphone. So you see the transparent bit, which is just fixating it in the ear, and the blue bit is emitting ultrasound. So basically, the ultrasound is activating parts of the vagus nerve that is situated around the ear. And this vagus nerve stimulation is then activating the parasympathetic system. So what you see is that you have a wide range of benefits. People sleep better. There's one study just coming out where we show reduced anxiety. So you use that device for 5 minutes a day. And then after a week, anxiety is already down by more than 30%, and then down by more than 50% after a couple of weeks. And 85% of participants are responding to this intervention. So this is something that is already on the market. You can buy it online. It's marketed as a wellness device because it doesn't have FDA approval. So it's not. It didn't go through randomized clinical trials. It didn't go through the whole FDA approval process. But we know that it's working for anxiety. So if you want, you can already buy it online.
What innovations do you see in this focused ultrasound that you're talking about, possibly maybe in the next 3 to 5 years?
So I think the next 3 to 5 years, there will still be a lot of basic research and a lot of pilot tests in clinical studies and in patients. But beyond the next 5 years, I would think that this will change clinical practice. So there was one psychiatrist who basically said that in the future, every psychiatrist will have a sofa in his office and a brain stimulation device. So the idea is that the future is changing, and people will not just prescribe pharmaceutical drugs, but they will basically have the ability to use brain stimulation in order to get the brain into a different state, in order to have some immediate and long-term changes in the brain to help patients. And I think this is something that will happen in the next 5 to 10 years. For magnetic stimulation, this is already something that is used in the UK in the National Healthcare Service. So basically, you already have hospital units that are using magnetic stimulation for depression. So there was a large study published in Nature Medicine by colleagues here in Nottingham, and they were looking at magnetic stimulation for depression and looking at the long-term benefits of that. So you can basically see benefits over more than half a year in some patients, that you really have long-term changes in the system, and you don't have the side effects that you would see for pharmaceutical drugs. So the one thing when you run a drug trial for pharmaceutical drugs is that many patients are dropping out because of the side effects. But for brain stimulation, the patients are so eager that they basically come back for more brain stimulation. So they basically finish the whole series of brain stimulation treatments until the last session. And you don't see that in drug trials due to the side effects.
Right. So very interesting. No side effects. Do you see this replacing pharmaceuticals in the next few years?
On the long term, I think there's a real possibility, because what you see for pharmaceutical drugs is that you take the pill and then the pharmaceutical goes into the whole body, not just into the brain. Okay, so it goes through the blood, it goes to the heart, it goes to the lung, it goes to your whole body, and it goes to the brain. And when it goes to the brain, it also goes to the whole brain, not just the area that is involved in a certain disease. Okay, so if you have dementia, initially you have hippocampus involved. For Parkinson's disease, you have other subcortical regions, but the pharmaceutical drug just goes everywhere. And that's a huge problem. So you are not only having the desired effect at your target area, but you also have other effects in the rest of the brain and in the rest of the whole body. So this is one reason why, for example, for antipsychotics and for other medicine, you not only have beneficial effects, stopping hallucinations, but you also have obesity. You have lots of side effects. You even have some drugs that make you more suicidal. So you have a lot of unwanted side effects because the drug just goes all over the place.
Right. Professor, you are going after schizophrenia, depression, and Tourette syndrome. Are you saying that in the next 5 years or 10 years, this focused ultrasound neuromodulation could completely treat these patients? Are you saying that? And besides schizophrenia, depression, and Tourette syndrome, what are the other areas are there you think where this focused ultrasound will really help?
Yes. So starting with the first one. So initially we are focusing on areas where the drugs don't work. Okay, so you already have antipsychotics for hallucinations, but we are looking at cognitive impairment in patients. So people have reduced working memory and reduced cognitive performance. And this is affecting the ability of patients to get employment and to do everyday tasks. So it's really affecting the quality of life of patients. So we're basically looking at parts of the condition that are not affected by drugs at all at the moment, and we try to improve them in the next 5 years in our program of research. But on the long term, potentially our approach could be used to areas where drugs are currently working and then lead to the same effect that drugs are having without having the side effect that drugs are having. So I think on the long term, the idea is to have broader applications. And there are initiatives worldwide to try focused ultrasound for a range of conditions. So it's not just depression and schizophrenia and dementia. People are looking at it in terms of head injury. So there were animal studies showing that if you use ultrasound stimulation after a lesion, after some injury of the brain, you get a much better recovery. So this is something that would be interesting to test in human participants who have a stroke or who have a traumatic brain injury, whether this also works. So there's really a wide range of conditions that are currently being tested.
Professor, can you talk about your work on predicting epilepsy surgery using connectome analysis?
So we had several papers where we tried to predict whether surgery in epilepsy patients works. So around 1/3 of epilepsy patients don't react to pharmaceutical drugs, so they still have ongoing seizures. And because of that, surgery is kind of the last resort, the last option for them. But unfortunately, when epilepsy patients undergo surgery, in 1/3 of the cases, the surgery doesn't work. So people still have epileptic seizures afterwards. We were trying to predict what is going on and basically predicting whether surgery works or not. So we had one article many years ago trying to look at how the brain is connected, looking at fiber tract connections between brain regions and trying to predict whether patients are surgery free or not. And that works quite well. So we can look at this connectivity between regions. We had a more recent article where we also look at some connectivity within regions. So it's not the microconnectome level I mentioned earlier. It's not like connections between individual nerve cells, but it is connections between smaller parts of regions. And if we look at this, we get an even better prediction accuracy of predicting whether surgery works or not. And then we had one paper in 2017 in Brain where we predict whether surgery works or not based on implanted electrodes in epilepsy patients. So what you do for those patients is that you implant a set of electrodes under the skull, And you measure what is going on in the brain. And in particular, you measure some seizures happening in those patients. And based on that, you determine which part of the brain should be removed. So we had this kind of information, and we were looking at similar activity patterns between electrodes. So we were reconstructing functional connectivity between electrodes in those recordings. And we were reconstructing that based on the times where they didn't have a seizure. So it's basically just baseline activity in the brain without any seizure going on. So we were reconstructing this network, and we were then running computer simulations and models on the network, predicting what is the most likely starting point. And the interesting bit is that whenever the starting point that we predicted was the same point that surgeons were taking out, the patient was free of epileptic seizures. But if the surgeons were taking out a point that was different from our prediction, then the patient still had epileptic seizures. So we basically could predict whether surgery is successful or not. But we also have this model trying to predict which part of the brain should have been removed. So which area is most likely to be the starting point. And this is something that is being taken further by Professor Peter Taylor at Newcastle University, who was at that point working in my lab. So he's basically leading a large project trying to find better ways to predict how to treat epilepsy.
Right. Professor, you've done some work on brain folding. Can you explain this gyrification, your research and findings, and how might these findings inform neurodevelopment disorder research?
Yes. So that was work by Yujiang Wang, who's also professor at Newcastle University now and was working in my lab at that point. So she was basically looking at the shape of the brain. You can look at the surface of the brain, and the surface is basically folded. And the degree of folding is called gyrification. So if you have very strong gyrification, you have lots of folding. If you have no gyrification, which is called lissencephalic, the whole surface is just flat. So you find a flat surface for other animals. So you find it for marmoset monkeys, for example. But for humans and for rhesus monkeys, you basically have this folded structure. So we know that the folding is changing for certain brain diseases. So for many neurodevelopmental disorders that originate in childhood, there's a bit less folding going on, which basically means that there might be some changes during development. So we had one paper in PNAS where we basically look at the folding pattern and we observe scaling laws. We find some regularities. But we also see that if we look at dementia patients, the scaling law is slightly different. So it's like a slightly different shape of the scaling law. So it's one other difference we see for brain diseases compared to the normal organization of the system.
Professor, you are the chair of Neuroinformatics UK, and you are pushing for AI-driven analysis of MRI, EEG, and ECOG data. How do you think this is going to reshape our clinical neuroscience?
So basically, Neuroinformatics UK is a network of 600 researchers in the UK and also abroad. We have different areas within the network, so clinical, experimental, and computational work. And the idea is really to bring people together. Most of the network is more about biological brains rather than artificial neural networks. And the idea of neuroinformatics is to organize and analyze the data that we get. It could be EEG data, it could be MRI data. And there are ongoing studies showing that you get better results if you take into account how the brain is organized. So you can use MRI information to plan brain stimulation experiments, whether it's magnetic or with ultrasound, in order to make sure that you hit the right target. You can use connectome information to get a stratification of patients. So we can distinguish healthy participants from depression patients, for example. But using that information, it might be possible to get subtypes of depression patients. And those subtypes might help you to decide which pharmaceutical drug is working or which other intervention is working. So it's basically looking at data-driven interventions rather than only looking at the metadata. So what is the the patient history, what is the age and gender of the patient, and what are symptoms and so on, to also look at how the brain is organized and use that as a starting point to influence what kind of intervention you would see for that patient.
Right. Professor, what are your thoughts on this global race of, where some of the largest companies are trying to build generally purpose machines, because today we have these statistical models. This is data in, data out. You feed in a whole lot of data, and then you have it spits out a lot of data, which I don't think is the right approach to building generally purpose aware machines. You have been invested in neuroscience, neuroinformatics, and I believe that Neuroinformatics UK is also setting up like a technological branch, trying to kind of understand the intersection between neuroinformatics and neuromorphic computings and brain inspired algorithms. Can you talk about a little bit about that, and how do you think that's going to play a role in building possibly what we call this generally purpose aware agents?
Yes. So at the moment, there's still differences between what is done in artificial intelligence and deep learning and how the brain is organized. One researcher in Manchester, Steve Ferber, so he was the architect of the ARM processor. He was basically building a neuromorphic computing that is closer to how the brain is organized. So you basically have those individual chips, and whenever a neuron is firing, the firing is transmitted as a packet. So like on the internet, you have different packets, 64 bytes, that are transmitted over the network. So in a similar way, for the Spinnaker system at Manchester University, you have action potentials that are distributed as messages through the network. So this is a lot closer to how the human brain is working. So they have a system there where they can simulate 1 billion neurons. So they're a very large scale system. And then the challenge is how to organize the network. So you want to have the same network architecture as in the human brain. But I think that's a challenge in the future. So I think there is a limit to the architecture that you have in artificial neural networks at the moment. So all those GPUs or all those specialized machine learning chips and how the brain is organized. And one difference is the modular organization I was mentioning earlier. But the other difference is that the human brain is organized in space. And because it's organized in space, you have delays for how long it takes messages to go between different brain regions. And we know that those delays can be very beneficial. So we had one paper 2 years ago where we can basically show that the brain is optimized in a certain way to optimize delays in the system to make sure that there is more fluctuation in brain activity and less overall synchronization. So if you have too much overall synchronization, it's like an epileptic seizure. Everything is active at the same time. So that's not good. So you don't want too much synchronization in your system. And higher fluctuation is beneficial because you can easily jump from one brain state to another. So you can easily go from one thought to another thought. And we see for dementia patients that they are stuck in their thoughts for longer. So if you look at EEG states, they're just stuck longer in a state before they can jump to another state. So this delay organization is very important for proper brain functioning. And delay is something that you don't really have in computer system. Everything just goes from one computer step into the next step. You don't have delays of 5 to 10 to 20 steps in your system. So I think there were some limitations in the artificial neural networks that we have at the moment.
Yeah. Professor, are there any peers that you look up to who have taken this approach of focused ultrasound neuromodulation and which has shown really real world benefits?
There are many researchers in the area that have pioneered ultrasound, and it's really just taking off in the last 10 years. So I was participating in the first ultrasound brain stimulation meeting in 2019 at Oxford University. And at that point, there were only 100 researchers. And I would say 80% of them were just interested in the topic. They didn't do any research yet. And in the last ultrasound meeting, I think there were around 300 participants. So the field is really exploding, and there are more and more labs worldwide that have the equipment. So for the main equipment that people are using. There are now more than 100 labs worldwide that have the equipment. So there are more and more labs looking into this and running experiments.
So my last question to you, it took, I think somewhere around maybe 13 years, maybe I could be completely wrong, to, create the human genome project, but even after creating that, we still grappling, we don't completely understand human genome. You mentioned that, maybe in another 40 years we'll be able to possibly create the entire map, structural and functional, of a human brain. What's your intuition? When do we get this map? And maybe paint a picture of that world. What would that world look like? If we have a complete map of a human brain, do you think that we would be able to take these neurological diseases, out of the system completely? And what would be those first few diseases which could be completely erased?
Yes, yes. So first of all, I don't think we need the complete map in order to make progress for diseases. If we have the complete map, the disadvantage is that, as Olaf Sporn said, if you have a detailed map of the human brain, you have the map of one human brain, and that person is dead. um, because we have to slice up the brain and do the electron microscopy and so on. And we know that each human brain is different. It's like a fingerprint, every brain has a different organization. So even in the best case scenario, after 40 years, you reconstruct one brain, all you have is one brain. So I think the benefit of reconstructing one brain might be uploading in the future. So uploading your brain to the computer. So you don't mind that you're dead because you live on in the computer, and then this will be the market. So for that, this reconstructing the whole brain will be useful. One challenge for uploading that people don't take into account at the moment is that you have an error when you reconstruct any system, right? You have a measurement error. So in biology, people always say there's at least 20% error in your measurement. So let's say you do a measurement of the human brain and you have an error, which basically means that there are errors in your connections. So maybe some connections are not there, or some weights of connections are not realistic. And this could influence the activity in the reconstructed computer system. To some extent, you get around that by allowing for learning in your networks. If you increase the learning at the moment when you're uploading, you can self-correct for some of the errors. So let's say you have an orchestra. You have 40 people playing in an orchestra, and one person is just out of tune. And if one person is out of tune, well, basically that person can look at the rest of the orchestra and then get back into tune. So in a similar way, if you allow learning, then even though if one neuron is slightly differently connected, it can recognize what is going on in the network and then changing connection weights in order to function in a way it has functioned properly. So to some extent, you can correct for those errors, as long as those errors are not too strong. So if you have too much error in your system, then you can't correct for it anymore. The second source of error when uploading is that you're not able to reconstruct the whole human brain at the same time. So you have this anime, I forgot the name of the anime series. So there's one series where you basically have uploading, and you see a laser that is basically scanning through the whole brain, and then the problem is that you don't get the brain at the same time, right? I mean, you have one slice of the brain that you're scanning, and then maybe 5 seconds later, you have another part of the brain. And 5 seconds is like an eternity, and to some extent, in terms of what is going on in the brain. So you basically get activity or you get information at different time points. And again, this is introducing error in terms of reconstructing the brain. So there were some challenges to uploading, but I think that would be a big result if you reconstruct the whole human brain. But I think for diseases, we can already get benefits much earlier. We don't need to reconstruct the human brain to the single cell level.
Right. Professor, thank you so much for taking time and being part of the podcast. Wish you and the entire team the very best. Yes, I think we need more brave researchers such as yourself who's zigging while the world is zagging, because yes, I mean, you pointed out that, the drugs, pharmaceutical industry, I mean, it's done as good, but it's got so many side effects. There is these invasive approaches, which has got its inherent problems. Obviously, it's expensive, and then nobody would want their brain to be drilled. And these three invasive, non-invasive approaches, magnetic, electrical, and out of that, ultrasound. Who would have thought that sound would have plays such a big role? And here we are, just kind of trying to kind of figure out what the future would be. So I hope that I mean, there is more people joining in, there's more collaboration, there's more understanding, there's more evolution of this tech stack. And I hope that this focused ultrasound neuromodulation plays a huge role in the future of humanity, to take away, I think, the biggest problem of humanity, which is this neurological disorders, your dementia, your schizophrenia, and so many other things. If we kind of take that out of the system, I think there'll be so many people who will be given a second chance to live, and live in a world and do what they actually want to be. So please keep on doing what you're doing. Wish you the very best. And to my listeners, if you like what you see in here, then please press the subscribe button. Until next time, so you get. Thank you, Professor. Really, really appreciate this. Thank you.
Thanks for having me.